That project is a worldwide project. It started by scientists and researchers, governments coming together to look at mapping.
In 2019, geneticists analyzed DNA from over 250,000 people across every inhabited continent on Earth.
We're trying to understand what accounts for the variability in mutation rates and how that changes longitudinally in people with age.
The goal was straightforward. Map the genetic age of human populations and see what the data showed.
Humans around the world have much in common, but also enormous diversity. Some of the differences between each of us come from our environment and life experiences.
What came back upended assumptions that had been sitting comfortably in textbooks for decades. The population that ranked oldest was not the one anyone expected. The study that started the debate. In 2019, a genetics research team set out to do something that had never been done at quite this scale before. They collected and analyzed DNA from more than 250,000 people, drawn from populations across every inhabited continent on Earth. The goal was to measure genetic age across human populations globally and produce a ranking based on what the data actually showed, rather than what existing models predicted. The results were not what the researchers expected. They were not what the broader scientific community expected either. And when the findings made their way out of academic journals and into public discussion, they sparked debates that are still running today across genetics, anthropology, and the wider conversation about human origins and what our DNA actually tells us about where we came from. The central conclusion the study reached was direct and uncomfortable for anyone attached to the existing framework.
We knew there was 3 million bases, that's the letters of code, but we didn't know that there was 20,000 genes, what they did, how they worked together.
The conventional understanding of human ancestry, the version taught in schools and repeated in popular science writing for generations, was described by the researchers themselves as fundamentally incomplete. Not wrong in every detail, but missing enough that the picture it presented was significantly different from what the full data revealed. To understand why the findings landed the way they did, it helps to start with what genetic age actually means, because it is not the same thing as the age of a civilization or the age of a culture, and the distinction matters enormously for reading the results correctly.
Scientists would say, why would you sequence the whole genome? We're only interested in the interesting bits.
What genetic age actually measures. Genetic age has nothing to do with how old a culture is, how long a civilization has existed, or how developed a society appears by any external measure. It is a measure of genetic diversity, specifically the number of mutations that have accumulated in a population's DNA over tens of thousands of years. Here is how it works. Every time a cell copies its DNA, small errors occur. These errors are called mutations, and they are almost always harmless. They accumulate across generations the way sediment layers accumulate at the bottom of a lake, building up steadily over time into a readable record. More mutations mean a population has existed in a particular place for longer. The depth of the sediment tells you how long the lake has been filling. Within a population, researchers measure what is called nucleotide diversity, which is the degree of genetic difference between individuals within that group. Higher nucleotide diversity means the population has had more time to accumulate those differences. Lower nucleotide diversity means the population is younger in genetic terms, or that it went through a period where its numbers dropped so sharply that much of its diversity was lost. That second possibility is what geneticists call a bottleneck. When a population shrinks dramatically, whether through disease, famine, climate disaster, or migration, it loses a large portion of its accumulated genetic variation. The survivors carry only a fraction of what the original population held. When the population grows again, it grows from that reduced foundation. Migration is one of the most reliable ways to create a bottleneck. When a group leaves its original home and moves somewhere new, it takes only a subset of its community's genetic diversity with it. The people who stayed behind keep the full range. The people who left carry a reduced version. that reduction is permanent and traceable. Geneticists can follow the trail of reduced diversity across continents like a breadcrumb path, mapping where groups went and roughly when they left. All of those breadcrumb trails, when followed back far enough, lead to the same place. Every piece of genetic evidence gathered from every human population on Earth points toward a single origin point. That origin point is Africa. How the science evolved to make this possible. Before the 1980s, understanding human ancestry meant working with physical evidence, bones, stone tools, cave paintings, and the geographical distribution of artifacts. These were valuable, but limited. They told researchers roughly what happened and roughly when, but the picture they produced was blurry and easy to dispute. In the 1980s, a breakthrough changed the field. Scientists developed the ability to sequence mitochondrial DNA, the genetic material passed exclusively from mother to child, unchanged except by mutation. Because mitochondrial DNA is inherited only through the maternal line, it functions as a precise clock for tracing ancestry back through female lineages across thousands of generations. It was the first genetic tool that allowed researchers to map human migration with real accuracy. In the 2000s, whole genome sequencing pushed the capability much further. Instead of reading one specific strand of inherited material, researchers could now compare millions of genetic markers across an individual's entire genome, all 3 billion base pairs of it. Applied to large populations simultaneously, this produced a level of detail that the earlier methods could not approach. The 2019 study used whole genome sequencing across its full sample of over 250,000 individuals. It measured nucleotide diversity within each population group and built a ranking from lowest to highest. The results of that ranking are what generated the debate that followed. The populations with the youngest genetic age. Starting at the lower end of the genetic diversity ranking helps explain the overall pattern. The group with the lowest genetic diversity is the indigenous populations of the Americas. Their ancestors crossed the Bering Strait about 15,000 to 20,000 years ago. At that time, sea levels were lower, so people could walk across. This was the most recent major human migration. The group that made the journey was very small. Because of this, a genetic bottleneck happened. A small group means less genetic variation. As a result, indigenous populations of North and South America have the lowest genetic diversity. This is not related to their abilities or cultures. It is only because of when and how their ancestors migrated. East Asian populations have slightly more genetic diversity. Their ancestors left Africa around 50,000 to 60,000 years ago. They traveled through the Middle East and South Asia before reaching East Asia about 40,000 years ago. This journey also involved a small founding group. So, a bottleneck effect also happened here. Over time, East Asian populations developed certain traits. These include differences in diet, hair type, and even earwax type. These changes came from adapting to their environment over many years. European populations have a similar level of diversity to East Asians. Their ancestors also left Africa around the same time. They moved through the Middle East and then into Europe. Europeans have a small amount of Neanderthal DNA. This happened because early humans met and mixed with Neanderthals. Some visible traits in Europeans include lighter skin. This helps the body produce vitamin D in places with less sunlight. Many Europeans can also digest milk as adults. Middle Eastern populations have higher genetic diversity. This region was a meeting point for many migrating groups. People from different areas passed through and mixed over time. This increased genetic variation. South Asian populations have even higher diversity. Many groups stayed in the region for a long time. They did not move as far as others. Because of this, they kept more genetic variation from early human populations. Africa and the populations that stayed. Moving into the upper range of the genetic diversity ranking means moving into Africa, which holds the oldest and most diverse human populations on Earth by a margin that is not close. This is what the migration model predicts, and it is what the data confirms. The populations that never left the continent where humanity originated, or that left only briefly and returned, carry the deepest accumulation of genetic variation. They did not experience the bottleneck effects of long-distance migration. They did not found new populations from small groups. They stayed, and their DNA accumulated mutations across longer periods of uninterrupted residence than any population that moved elsewhere. East African populations, particularly those in Ethiopia, Kenya, and Tanzania, sit near the top of the global diversity ranking. The region contains some of the earliest known fossil evidence of anatomically modern Homo sapiens. Groups like the Hadza of Tanzania show genetic evidence of over 100,000 years of continuous habitation in the same region, a depth of rootedness that produces a corresponding depth of genetic diversity. The Hadza are a small community, numbering only in the hundreds of individuals, but their genetic profile is a window into a period of human history that most other populations have moved too far from to retain clearly. And then there is the finding at the very top of the ranking, the result that generated the most attention when the study was published, and that has continued to generate discussion since. The population that ranked first. The San people of southern Africa, also known as the Khoisan, ranked as the most genetically diverse human population on Earth. Their nucleotide diversity is approximately 0.1%. For comparison, European populations show nucleotide diversity of approximately 0.06 to 0.07%. That gap is larger than it looks in percentage terms. When translated into actual genetic differences between two randomly selected individuals from each population, it means that two San individuals selected at random differ from each other at roughly 3 million points across their genomes. Two Europeans selected at random differ at roughly 2 million points. That is not a small difference in genetic terms. It represents tens of thousands of years of additional accumulated variation, and it places the San in a category that is distinct from every other human population the study examined. The San people's ancestors have lived in southern Africa for at least 100,000 years, and some estimates based on the most ancient lineages within their genetic profile push that figure considerably higher. Their genetic lineages diverged from those of other human populations before the major migrations out of Africa even began. In the family tree of human genetics, the San represent branches that split off earlier than any others currently identified. They carry haplogroups, meaning specific inherited genetic signatures that exist nowhere else on Earth. Their mitochondrial haplogroup is called L0, and it is the oldest known human mitochondrial haplogroup. Every other mitochondrial haplogroup that exists in any human population anywhere on Earth descends from lineages that came after L0. The San are not a branch on the human family tree. They are closer to the trunk than anyone else currently alive. The evidence beneath the evidence. The genetic story of the San people does not stop at nucleotide diversity or mitochondrial haplogroups. It goes deeper, and the deeper it goes, the more remarkable it becomes. In 2013, researchers studying Y chromosome data, the genetic material passed exclusively from father to son, identified a haplogroup called a 00. The estimated date of this lineage's split from all other human Y chromosome lineages was calculated at somewhere between 240,000 and 580,000 years ago. To put that in context, anatomically modern Homo sapiens, as a recognized species, is generally dated to approximately 300,000 years ago. The A00 haplogroup's split point sits at or before the emergence of our species as a distinct biological form. It predates the major expansions and migrations that produced every other human population currently living. This haplogroup is found almost exclusively within populations connected to the San and closely related groups in southern Africa. Its presence in their genetic profile means that the San carry lineages stretching back to a period so early that the boundaries between Homo sapiens and its immediate predecessors were not yet clearly drawn. There is also evidence, still being studied carefully, that San DNA may carry traces of interbreeding with archaic human populations for whom no fossil record has yet been found. The signals in the genetic data suggest contact with relatives that left no bones in any known archaeological site, but left a faint mark in the genomes of the people who encountered them. The San may carry a living record of human relatives who are otherwise entirely invisible to science. Taken together, these layers make San DNA something that geneticists have described as a living archive of human evolution, a record that cannot be recovered from any other currently living population. Migration as a series of reductions. One of the things the 2019 study clarified most effectively was the picture of how migration out of Africa actually worked. The simple version of the story, one large wave of modern humans leaving Africa around 60,000 years ago and spreading across the world, does not fit the full genetic data. The real picture involves multiple waves, with different outcomes for different groups. The first major wave that left lasting genetic signatures departed approximately 60,000 to 70,000 years ago. This is the founding migration for non-African populations worldwide. But there were earlier attempts. Groups left Africa approximately 100,000 years ago, reached various parts of the Middle East and possibly further, and then disappeared. They left no lasting genetic signature in any living population. They went extinct, likely because they moved into environments they were not yet well adapted to, or encountered conditions they could not survive long enough to establish stable communities. The African populations that did not migrate avoided those extinction events by remaining in the environments where Homo sapiens had already spent hundreds of thousands of years adapting. The continent that produced the species was also the one that provided the most reliable conditions for surviving in it. Staying home was not a failure to migrate. It was a condition of deep continuity that produced the genetic diversity the 2019 study found at the top of its rankings. Each subsequent migration, from Africa to the Middle East, from the Middle East to South Asia, from South Asia to East Asia, from Northeast Asia to the Americas, reduced the genetic diversity carried by the migrating group. Each new founding population was a subset of what came before. The genetic differences between populations living today are not measures of quality or capability. They are measures of distance from the origin and the number of bottlenecks a lineage passed through on the way to where it ended up. Why this matters for medicine? The implications of this ranking are not limited to academic discussions about human prehistory. They have direct practical consequences for how medicine is practiced and how pharmaceutical research is conducted. The overwhelming majority of large-scale pharmaceutical trials over the past several decades have used populations of European ancestry as their primary subjects. Drugs are tested, dosages are calibrated, and safety profiles are established using data drawn mostly from one relatively narrow slice of human genetic diversity. Those drugs are then deployed globally across populations whose underlying genetic profiles may differ in ways that affect how the drug behaves. This is not a theoretical concern. Documented differences in drug metabolism, dosage requirements, and side effect profiles across different population groups reflect real underlying genetic variation. A drug calibrated for one genetic background may work differently or produce different risks in a population with a substantially different profile. The San people and other high-diversity African populations are particularly valuable in this context. Their genetic variation contains combinations and sequences that do not appear in lower diversity populations. Within that variation may be clues to disease resistance, longevity, and metabolic processes that current medicine has not yet been able to study simply because the research has not been done with adequate representation of these groups. The push within genetics and pharmaceutical research for more inclusive study populations is partly driven by fairness and partly by straightforward scientific logic. If the goal is to understand how human biology works, studying only a fraction of human genetic diversity produces an incomplete picture. The populations that carry the oldest and most varied genetic profiles are the ones whose inclusion would add the most to that picture. The ethical dimension of this conversation carries its own weight. The populations with the oldest and most scientifically valuable DNA are frequently the same populations that have experienced the most severe effects of colonialism, displacement, and systemic harm over the past several centuries. The San people of southern Africa have faced land dispossession, forced relocation, and systematic marginalization across generations. The fact that their genetic heritage is now recognized as the deepest in the human family tree sits in complicated relationship with how their communities have been treated by the modern world. Researchers working in this field have been direct about the need for careful ethical frameworks governing how this DNA is studied, who benefits from the research, and how communities are involved in decisions about their own genetic information. The misconceptions worth clearing up. The results of the 2019 study were misread in certain public discussions in ways that the researchers themselves pushed back against directly. And those misreadings are worth addressing plainly. Genetic age does not measure intelligence. It does not measure capability, creativity, social complexity, or any other quality that might be used to rank human groups in terms of value or worth. It measures one thing: how long a population has accumulated genetic mutations in a particular region, shaped by migration history and the bottleneck effects that migration produces. Lower genetic diversity does not mean a population is less advanced. The indigenous populations of the Americas, who rank at the bottom of the genetic diversity scale, built Tenochtitlan, Machu Picchu, and Cahokia. The Aztec, Maya, and Inca civilizations were not primitive. Their ranking reflects the recency of the Bering Land Bridge crossing and the small size of the founding group that made it, nothing else. The San people are not living fossils. They are modern human beings living modern lives, making contemporary choices, and navigating a present-day world. Their genetic profile is ancient. Their existence is current. These are not the same thing, and treating the first as a description of the second is a category error that the researchers explicitly warned against. The rankings in this study are a map of migration history and geographic circumstance. They are not a hierarchy of human groups arranged by any meaningful measure of human value. What the full picture shows. Pull back far enough, and what the 2019 study produced is not a ranking in the competitive sense, but a map of connection. Every human population alive today, from the San people of southern Africa, to the indigenous communities of the Arctic Circle, to the populations of East Asia and Europe and the Americas, traces its ancestry back to the same origin point on the same continent. The differences between populations at the top and bottom of the genetic diversity ranking are differences of time and geography. They reflect where groups went, when they left, and how many bottlenecks their lineages passed through along the way. The deeper the diversity, the longer the unbroken thread of residence in the land where the species first emerged. The San people carry the deepest thread of any group currently living. Their ancestors were present in southern Africa before the migrations that founded every other human population on Earth had begun. Their genetic lineages were already ancient when the groups that would eventually reach Europe, Asia, and the Americas were still part of the same African community. That is what the data says. The oldest human DNA currently walking the Earth belongs to a community living in southern Africa, in lands their ancestors have occupied for longer than any other human group anywhere on the planet. The study did not answer every question it raised. The genetics of deep human prehistory still contain large areas of uncertainty. and the picture will continue to be refined as more populations are studied and sequencing technology improves. But the broad conclusion, the one that the researchers described as challenging the fundamental assumptions of the conventional model, held up clearly in the data. Humanity's deepest roots are not lost to history. They are alive, walking landscapes that human feet have crossed for longer than anywhere else on Earth. New genetic research challenges old ideas about human origins. It shows our deepest roots trace back to southern Africa, linking all people today to a shared beginning. If you want more clear, evidence-based stories like this, subscribe now and stay with us for the next discovery coming very soon.